Hello, my dear students. Hope you are doing well. We have discussed in the previous class the molecular biology and genetics, which is a long subject in which very important topics are there. The DNA, the genetic material, and the genetic material is the center point in the whole cell. The centrally organized, all the processes which are going to be done in the cell will be organized by the genetic material and truly transforms the genetic characters into the next pedigree. And how exactly this DNA or otherwise genetic material is, and we learned already, that is DNA a double helical, right-handed double helical structure with 4 important nucleotides, 2 of them purines, 2 are pyrimidines, adenine, guanine, thymine, and cytosine. Always the base pair, the fundamental, and adenine always pairs with the thymine, and guanine with the always pairs with the cytosine. The most important base pair rule, always it follows. And the second important nucleic acid is the important one is the main one is the DNA, and the associated the one is the RNAs, and ultimately both orchestra, the DNA and RNA, will finally orchestrate to produce the protein, and that protein will be the main principle behind various characters of individual organisms or individual beings. The color of the hair, the color of the eyes, the height of an individual, the weight of an individual, all these things. Well, and we have learned how a DNA molecule, when a mitosis is going on, can be truly with a high fidelity, high trust, without having any single mistake. How many nucleotides we have learned in a particular human nucleus? At least about 0.7 trillion nucleotides, 700 billion nucleotides are there. 10 to the power of 10, almost 7 into 10 to the power of 10 nucleotides are all there, and so many of them are arranged in a specific order, and which order shall not change. If changes, at times there are going to be grave consequences, death, disease, or at times lucky can escape the problems. Well, with this, what we have learned previously, we also learned in details a process called as replication. A DNA of a cell truly producing another such molecule, and then that will be distributed to the cell which is going to be dividing, and then both of them will be having 2 nucleus individually. This is what is the replication. We learned that it is semi-conservative. 2 strands of the parent is equally shared into the 2 daughters, and then 1 complementary strand in each are being synthesized. What is the important enzyme in the replication? Can you recall back? DNA polymerase. DNA polymerase. DNA polymerases are, the nature of them are a bit different from that of prokaryotes to that of the eukaryotes. What are prokaryotes? Without nucleus. Doesn't mean that they don't have DNA. Their DNA is present in the cytosol organizedly. Nucleide, that is what is called as. Whereas in the eukaryotes, this long huge DNA is being folded, is being wrapped around the protein's histones, and then have secondary conformation, tertiary conformations, and then finally chromatids, and then chromosomes, and then diffuse nuclear mass. This is what is the nucleus. So now this is how the cell gets another genetic material, that is the DNA, after replication. And the main important enzyme for synthesis of the DNA second strands, the DNA polymerase. There are few differences between prokaryotes and eukaryotes. Whatever it is, that is all being discussed. Then the later one is after replication, then the cell cycle also we have discussed. Now we are discussing the transcription and translation. Replication, transcription, translation. Replication, producing a total DNA molecule, another molecule from the parent to another daughter, otherwise one parent to two daughter molecules. This is the replication. Whereas transcription is in the process of producing proteins. DNA molecule, such a large, huge molecule, here and there, it does have the genes. Again, we come to the genetic concept. Initially, we do not know, but we used to have a philosophy that genes are there in the cell. And where are they? People doesn't know. But once DNA is known, and then later on, slowly, the DNA, one of the strand is supposed to be the gene of it. And it is confirmed now. And now the gene is present on either strands at a particular place. So the whole DNA, the complete DNA is not producing proteins completely. All the sequences are not going to produce proteins. Here and there, some sequences are there, some pieces of the strand, polynucleotide strands of a polynucleotide strand of the DNA molecule, the large huge DNA molecule, is otherwise a gene. That gene will be transcribed. The message that is being present on that gene, or the telegraphic message that is being present on that gene. In which manner it is present? In the manner of nucleotides. Adenine, thymine, adenine, guanine, cytosine, something like that. So, such a sequence is having a meaning that it is going to produce a protein. How does? Is what the great science has been revealed. And then the DNA, a piece of DNA is truly been producing another mobile polynucleotide strand, and that is otherwise called as a message of this gene is being there in this, and hence it is called as transcribed. So gene's message is being transcribed into another polynucleotide strand, and this is called as RNA, because here, while this is being formed, two important differences when compared to replication. And that is that in the replication, again, that newly synthesized strand will be associated with the parent strand, that is the template strand, and then both of them will together becomes another DNA molecule. Both of them will be reassociated. But whereas in here, a piece of the gene, the piece of the polynucleotide of DNA, that is the so-called gene, is being transcribed into another chain of polynucleotides. But here, they use us two very, very important differences. That is, the ribose is being used here, and then secondly, instead of a thymine, uracil is the again. So these are the two differences when this transcription is going on. So from the gene, that is the polynucleotide sequence of the DNA, another a piece, mobile piece, polynucleotide strand of RNAs of ribose and uracil containing polynucleotides are going to formed. But always it is dictated by the gene, that is the polynucleotide. And base pair rule is with the high trust it is being followed. And with that dictation, they one by one gets polymerized and produces a long chain. And this particular chain now of adenine instead of thymine uracil, guanine and cytosines. With these 4 bases, nucleotides are going to produce the RNA. And this RNA is mRNA. And there are other types of RNAs do exist. That types of RNAs we have already discussed in the previous class. The types of RNAs: mRNAs, rRNAs, pRNAs, and small RNAs. 4th category recently has evolved, which were not known in the smaller classes. Then this mRNA is truly takes the message or dictation from the gene, and how exactly the protein should be there, which amino acid is the first one, which is the second one, which is the third one. If that protein is having, suppose, 146 amino acids. So the sequence 1, 2, 3 up to 146, how it should be there. So these nucleotides are arranged in the gene and then are transcribed onto the mRNA. And this mRNA now dictates at the place of protein synthesis. Place of energy liberation, my dear students, where? Mitochondria. Like that, the place of protein synthesis, where? Ribosomes. So in the ribosomes, what is present? Some proteins, a complex of proteins, but mainly about 40% are the proteins and 60% again these RNA molecules which are being there in a massive manner. And these RNA molecules, you know it, called as they are present in the ribosomes, they are called as ribosomal RNA, and in the smaller classes also called as, because in the cytosol it is a soluble form, this is soluble RNA. So that is ribosomal. These RNAs are also synthesized from the signaling of somewhere in the DNA of the cell, that is in the nucleus. So some genes are there for the synthesis of RNAs also. Similarly, there are some genes for the synthesis of tRNA also. Then in the microbes, this specialty has not been developed much, but yet the synthesizing for the synthesis of RNA, and how are they synthesized? One nucleotide is being tagged to another, is being linked to another by a phosphodiester linkage, isn't it? So this phosphodiester linkages, when it is being done for the purpose of synthesis of DNA, we call it as DNA polymerase. Whereas now, what are we are synthesizing here? This is what is the cell is synthesizing here? RNAs are synthesized. So then RNAs are being synthesized. What shall we call that name? RNA polymerase. RNA polymerase. How can RNA polymerase synthesizes these strands with the dictation of DNA? Isn't it? So now DNA dependent RNA polymerase is a complete name. Got it? So now we have 2 very important enzymes for the synthesis of DNA. It is the DNA polymerase. For the synthesis of RNAs, and then to the purpose of protein synthesis, they go. And then the important enzyme needed is RNA polymerase. And then I also told you that it is the Crick who is the father of the DNA deciphering the model, and that man has given a great statement, central dogma of molecular biology. This central dogma of molecular biology is always a gene of a strand of the DNA is going to produce exactly the base pairing rule following a RNA molecule. And this, so synthesis of, and this is an mRNA molecule. The synthesis of mRNA from DNA is transcription. Come on, always, all of you, please note and don't forget. Transcription, synthesis of mRNA with the dictation of the gene, that is a strand from the DNA, is called as the transcription. And this mRNA, after the formation and few changes which we are going to discuss later, and then this one liberated from this has been liberated first initially in the nucleus, in the nucleoplasm, and it travels in the nucleoplasm, comes out of nucleopores and goes into the cytoplasm. In the cytoplasm, it reaches the ribosomes, where this is being interfaced in the ribosomes, or it is been aligned in the ribosomes, and then with the help of another of his brother molecule, tRNAs, and this tRNAs are having capability to bring the amino acids, catch and link them and bring the amino acids into the ribosomal assembly, where the mRNA tells which amino acid is the first one, which is the second one, which is the third one, and accordingly it identifies, the tRNA identifies, and that amino acid comes, and that will be linked in the ribosomes, and then one by one, 1, 2, 3, 4, 5, 146 amino acids will be linked, and a protein molecule will be completely, one amino acid is being linked to another amino acid by peptide linkage, and this linkage occurs in the ribosome, and the ribosomal proteins are having the capability to catch the amino acids, take out of the tRNA and linking it into the one to another, 1, 2, 3, one by one, first amino acid to the second, second to the third, and by the peptide linkage is being created by the ribosomal proteins with the help of ribosomal RNA, and at last a protein molecule is being completely synthesized. Once it's synthesized, the protein synthesis process for that molecule is complete and will be liberated into the cytosol. So this process in the ribosomal or in the ribosome, mRNA is producing a protein with the help of rRNAs and tRNAs and amino acids, and the whole thing, a bit of little energy, and finally a protein is synthesized. And once a protein is synthesized, and that dictates the characters of an individual, functions of an individual, enzyme capabilities of an enzyme, reactions of a particular processes in that cell and cell survives. This is the central dogma: is DNA produces mRNA, mRNA produces a protein. Always this goes from top to bottom, DNA to RNA to protein only. Reverse cannot be done is the initial concept of the Greek, and this is central dogma of molecular biology. But now there are exceptions we could be able to see. We found various viruses, and these viruses are not obeying this particular. They are having capability from an RNA. They have the capability to produce a DNA. Where? In a cell. Which cell? In the cell of the host. All RNA viruses are all dependent on some holo cells. Let it be a human cell. Let it be a bacterial cell. Let it be a yeast cell or a plant cell. They are all dependent. These viruses are all cannot independently survive. This is a different story. Now, as far as in our cell, a protein getting synthesized, always DNA to RNA transcription, RNA to protein synthesis translation. Each one is a subject by itself. Such a vast subject has been developed today. 50 years ago or 60 years ago, we did not know about these processes. And this has totally revolutionized our medicine. And now we are having capability to clone the individual, clone the organisms. We are able to produce a sheep from the sheep cells. And then like that, we are able to combat the cancers which we felt is impossible to do. And we are combating with the genetic diseases and all these things after having complete understanding of into the molecular biology. The highest possible science of medicine is how molecular biology. It explains every subject. Let it be medicine, let it be pathology, let it be microbiology, let it be physiology or anatomy, the molecular biology. Anyway, it is a part and parcel of the history. See, molecular biology, chemical pathology. So this is about central dogma, and after knowing it, transcription, producing RNA, and producing protein. And this will be acting and producing some characters. And this is the three types of RNA. And this is a linear RNA, mRNA, whereas ribosomal RNA is also a linear one with some secondary structures, but mainly present in the ribosomal assemblies in a cluster manner. And whereas tRNA needs a little bit of discussion. tRNA is clover leaf in nature. You see? Clover leaf in nature. Here it is having all these other RNAs are single-stranded, but even though this is also single-stranded, but when the complementary bases or does come across, and it is possible at this level, then they form double helices. But whereas here it is a single, whereas it is a single man, but the single manner. But here and there you can see double helix in this double-stranded complementarity, thus present in this tRNA. On one side, this is amino acid end, and this is here where these tRNAs, mind you, are not all are same. There are about 20 categories for each amino acid, and all of them are having some small difference, mainly at these 3 nucleotides. 1, 2, 3 nucleotides at this end. This is a codon end. This is a codon end. And this is a pseudouridine end, psi end. And this is a D end. And then this is a short arm. So 1 arms it has. This is 1 arm, 2, 3, 4 arms are there, and a small short arm are there. Depending upon the total number of nucleotides present, if number is small, about 4 to 6, then it is called type 1. And if it is about 15 to 20 nucleotides present in this small extra loop, and this is this small loop, and then it is called type 2. So tRNAs are having type 1 tRNAs, type 2 tRNAs, depending upon the extra loop containing few number of nucleotides, type 1, a little more, that is 15 to 20 type 2. Apart from that, these amino acid, alanine is bound by a specific species of tRNAs. Valine is bound by another species. So there are about a minimum 20 categories of them which are being classified into 2 types, type 1 and type 2. So transfer RNA, Foley is a scientist who has discovered this particular structure of this tRNA. Here it has unusual bases like pseudouridine, okay, like that. Yes. So such methyl pyrimidines, such ones are thus present in the primary structure of this tRNAs. Apart from that, there are what are called small RNAs. And these small RNAs, some are there in the nucleus, some are there in the cytosol. And in the nucleus, snRNAs, small nuclear RNAs, have the mRNA that is being liberated will be processed, and what does it process will be. And then there are others also there in the cytoplasm. They are called siRNAs or microRNAs. They have a regulation on the transcription and the translation processes. Look at both the places. They have a control over the processes. And the others are the SC, signal recognition particles. It is a tRNA process. And mind you, but the major ones, mRNA, rRNA, and tRNA, in human beings, in the mammals, and in these ones, or otherwise, the eukaryotic cells, the RNA is synthesized by RNA polymerase, I said. So, for all three different RNAs are produced by three different categories of RNA polymerases. mRNA is produced by RNA polymerase 2. rRNA is produced by RNA polymerase 1. tRNA is produced by RNA polymerase 3, grossly, but we have little overlapping functions. So, this you should remember. Eukaryotic DNA polymerase are three types: polymerase 1, 2, 3. Polymerase 2 is the one mainly transcription process purpose is being used. Whereas in the microbes, like the bacteria, the RNA polymerase is a single one, but it does have, because there are various processes to be done. For that purpose, it has 2 alpha peptides in that enzyme, and there are 2 beta peptides, and then omega peptide, and then associated with a factor called as Rho factor. And this holo one is called as DNA polymerase holo enzyme. For microbes, bacteria, eukaryotes, the RNA polymerase is a complex of various peptidyl fractions, 2 alphas, 2 betas, 1 omega, and a rho. So 6 fractions of protein thus totally acts together, produces an mRNA in case of bacteria, eukaryotes, whereas prokaryotes, whereas eukaryotes have RNA polymerase 1 produces the rRNA, RNA polymerase 2 produces mRNA, RNA polymerase 3 produces DNA. Remember, this is the latter one. We'll discuss later. This is all processes. The DNA, the double-stranded, while RNA is a single-stranded. Out of these 2 strands, only 1 strand is going to be the gene. Maybe it is present on this strand, or may be present in the other strand. Both the strands, they do contain genes. And in the total DNA molecule, about not much than 10% is really the gene portions, gene fractions. The other things are all not meaningless. They have a lot of functions to do, which are not known to us, but some of them which are known to us are the controllers, and they are the sensors. When a protein to be produced, when a protein to be stopped, and when a growth factor to be produced, when it is to be stopped, and the signal perceiving processes are all being hidden in that huge, large polynucleotides. Apart from the genes which are producing an mRNA and producing a protein, and which is functional. So now the main gene is being present on the, say, particular this strand, then this is a template strand. Basing on the message on the template strand CGG, exactly, totally following base pair rule. Base pair rule is the most important one, which cannot be deviated. If it is deviated, the living system is going to be deviated, and hence the nature cannot disobey the base pair rule. Base pair rule. So now, Here, say this is a gene which starts with initially a cytidine, guanine, adenine, so on like this. So when cytidine is there, then immediately it should be guanine. When guanine is there, cytidine must be there. And guanine must be there, cytosine must be there. Adenine is there, then normally if it is the DNA, thymine will come. But in RNAs, what should come? Uracil. Yeah, that is. So like this, taking dictation, using the RNA polymerase, it is going to produce polymerize one by one. But when it is, when gene is like this, but the process of transcription always from 5' to the 3' direction, and hence the template strand is read in the reverse, that is the 3' to 5'.